I want to take us through a journey on cassava genomics, and I'd like to preface this by saying that I work for a company that does not sell cassava. I started the cassava work before I joined Dow, and they've been kind enough to let me continue this work kind of on the side. Most of the work is done by the organizations on this slide. On the right-hand side are African partners, and on the bottom are our US partners. And I basically just give talks and write reports. So I'm a sequence guy, so every talk needs to have a slide about the rapidly reducing cost of sequencing, except I'm going to take this a slightly different direction. So this is a graphic that Illumina used to use to describe their flow cells. This is an old version of their flow cells. And each of these beads represent the oligos that are the base for the clusters of sequence on the flow cell. And this is a clip art that represents people on the planet. So I thought I'd take this a slightly different direction than we normally see these sequencing stats, and ask how many flow cells would it take to house the entire planet if every cluster was a person? And this is the answer. So just less than 5 flow cells will contain enough sequence reads to be 1 per person on the planet. And I usually give this talk to American audiences, and so it's kind of interesting when they see what small fraction of the world's population is in the US. And I work for an agricultural company, a seed company, and a major customer base are the US farmers. And so it's even more interesting when we point out that less than 2% of the US population live on farms. And that's that thin green line on the left-hand side of the slide. This is in very stark contrast to developing countries where over 2 billion people live on smallholder farms. So I think it's very important that we focus not only on crops that are critical for the developed world, but also that we use these same technologies to apply to crops that are not of interest to the big multinational companies usually. And cassava is one of those. Cassava is a particularly important crop in sub-Saharan Africa. It's also well known in this part of the world, but I'm going to focus most of the talk on sub-Saharan Africa and the role that cassava plays there. So for those of you who have not eaten cassava, this is it. It's a woody shrub with a very starchy root. The key relative to its use as a security crop in sub-Saharan Africa is that the edible part is below ground and there's no set harvest. So a farmer can leave this growing for pretty much as long as he wants and choose when he harvests, or when she harvests more typically, so that she can get the food at the right time of year. And some of the constraints that I'm going to talk about are really key to that decision. There are 3 main areas of research being focused on cassava improvement over and above traditional just yield improvement. The first is post-harvest loss, which is a real constraint in cassava because when you harvest a cassava plant, a root, there's small damage happens to the root. And this triggers a physiological deterioration of the root quality, such that within 48 hours it's pretty much unusable. So this really constrains the use of cassava in an economic way because you need infrastructure to be able to process it very rapidly. And so that kind of limits it to local consumption for the most part without the appropriate infrastructure. Another area is nutritional content. It's pretty low level of protein and micronutrients in the cassava root. And so there's been a lot of research trying to breed for improved nutritional content. The area that's most relevant for what I'm going to talk about is disease losses. And there are 2 major viral diseases that are key to understanding cassava constraints in sub-Saharan Africa. One is CMD, cassava mosaic disease, and one is CBSD, brown streak disease. So CMD is a Gemini virus. It's a DNA virus transmitted by whiteflies. And it's key in sub-Saharan Africa because there was a major outbreak of this disease in the late 80s that just had devastating impact on East African cassava production. And since then, there's been a concerted effort to use a particular locus that was discovered for resistance to CMD, and that is now very widespread in all of the breeding programs in Africa. As you see here, the leaf phenotype is extremely strong. It's very easy to tell your plant has CMD, and the root is a complete loss. This is a little bit contrast to brown streak disease, where the leaf phenotype is much more subtle, and a farmer may not know that his plant is diseased unless he's received some training or has some prior experience to CBSD. But the root is all necrotic when it's harvested and is unusable. Key to what I'm going to tell you later on in the talk is that CBSD was first identified as a virus disease in Tanzania in the 1930s. And what happened was that in response to this, a breeding program in Tanzania, the Amani breeding program, started deliberately breeding for resistance by using wild relatives of cassava. And they were very successful. They actually had some resistant lines that were able to survive the virus outbreak. But then the virus subsided, and the selective pressure that the breeders were able to use disappeared. And now we've lost the resistance that they had back in the 1930s. And so a lot of the efforts in CBSD research these days are trying to understand what they did in the 1930s and try and tap the remnants of that program to bring it back into modern day use. So I should say that the virus has now reemerged, and it is making West Africa very, very nervous because it's currently in East Africa and progressing slowly or rapidly, actually, into Central Africa. And so the biggest producer of cassava in the world is Nigeria, and they are very, very nervous that this virus will reach their borders. So, just to set up some of the species I'm going to talk about, I just want to give you an overview of the different terminologies we're going to use. So cassava is Manihot esculenta crantz, and I'll just refer to this as cassava. Wild cassava is a subspecies of that same species, but this time it's Flabellifolia. And then there is a true wild relative of a different species, and that's Manihot glasiovii, and that's the Sierra rubber tree. So these three taxonomies will come up when we talk about sequence data. There's also a tree cassava, and I don't know what the technical term for this is, but this is an ornamental tree found in East Africa, and it's believed to be an F1 hybrid between these two parents. So how can genomics help? We've heard all week about all of the technologies that genomics can bring to crop improvement. Specifically for cassava, we're faced with a situation where we're trying to figure out where to start from because there's very little to start. So we took a look at maize. Maize has a lot of resources, a lot of data resources that have been accumulated over decades, and more recently, a lot of genomic resources. And this has enabled applications to use those data. Applications on the right are more in the biotechnology realm, and breeding applications on the left. But if we're honest with ourselves about where cassava was about 4 years ago, 5 years ago, this is where we were. We had essentially zero data resources for cassava. There was not even a genetic map that had linkage group number that was close to the number of chromosomes. The densest genetic map had a few hundred markers on there. And so we were really starting from scratch. And so what we wanted to do was start building resources first. So the first resource is a draft genome. Back in 2009, we created a reasonably okay draft genome. And it kind of looked like this to a breeder. It's 12,000 pieces. And so for the ability to look at chromosome segments, this was not very useful. The ability to see what genes are in the genome is quite useful, but that's not what the breeders wanted to use. And so the next task is to try and translate that jigsaw puzzle into something a little more tangible, a chromosome scale assembly with greater contiguity. So one of the powerful tools for this is genetic maps, a dense genetic map. And cassava is not the organism you would pick if you wanted to do genetics. It's very difficult to produce large numbers of populations. Crosses are kind of tedious. You get one seed per attempt, and it's really difficult and hard work. So we polled the community, and we found 10 collaborators who had mapping populations that were willing to donate to a consortium that would put together a consensus map. We fully resequenced the parents of each of these populations, and we did GBS on the F1 individuals. And that gave us 1,700 individuals that we put into building individual maps and then a consensus map, which gave us then 22,000 markers and the right number of linkage groups compared to the chromosome number. This was recently published earlier this year. The very next thing we do with that is to anchor the sequence of that draft that we had. And now we have 90% of the genes anchored to a chromosome location. So we're beginning to get something that's pretty useful in a breeding context. And then just recently, we have taken a fresh look at the genome assembly using more up-to-date sequencing methods and have a slightly larger genome assembly. But this time, we've got 97% of the genes anchored to this, and the quality of the assembly is much higher. And we also have some PacBio data we're about to add later this year. So now, for the first time, we have a look, instead of the 12,000 pieces, we have chromosomes. And we can actually get a clear look at a whole genome duplication that we know existed in Cassava, or we suspected existed, but we didn't know in what state it was today. And it turns out that 5 of the pairs of chromosomes are almost exact collinearity with a duplicate. But the other chromosomes have gone through the complex of fission-fusion type rearrangements that give the chromosomes that we have today. So this would not be possible without the chromosome scale sequence. The very last data set I want to introduce is the resequencing of 50 plus accessions. And these were distributed between the cassavas, the wild cassavas, and the glasioviae. And you see here we've got African and South American cassavas, we've got flabellifolias in South America, and we have some Asian cassavas as well. So with all of that set up for the data sets, I want to give you some insights into what the sequence can sort of reveal. And the first is a kind of surprising from a genetic standpoint, and that is cassava is an outcrossing species, but we found very unexpected long stretches of homozygosity in every single line we looked at. These stretches are different in every line. So you see here, this is a plot of the density of heterozygosity. And you see, this is almost half a chromosome with very, very low heterozygosity. And this, again, is a whole chromosome arm with almost no heterozygote calls. And if we start adding up regions of the genome that are in homozygous state between the different accessions that we had sequenced, we actually find that 99% of the genes in cassava are homozygous in one of the accessions that we had sequenced or more. And this shouldn't happen in an outcrossing species. So how did this arrive? So we don't think there's any explanation here about selective pressure or selective sweeps because there's no one region that seems to have been focused on here. So the best explanation we can come up with is that this is just an evidence of recent selfing in cassava's history, even though it's an outcrossing species. And if you think about how cassava is cultivated, it kind of makes sense because we propagate cassava vegetatively. And so in a field that a farmer has, it's very likely that the plant that's going to receive the pollen for the outcross is a clone of the donor of the pollen. So a botanical outcross is actually a genetic self. And so we believe that's what's happened in the history of cassava quite a lot, is that we have quite a bit of selfing. But what we don't really understand is why these wild species would also show the same blocks of homozygosity. So that's something that's pretty new data. It's about a week old. So we haven't really gone in and really delved into what might be behind that. But it does have some implications. So cassava breeders often map traits with S1 populations. And this probably isn't a good idea. If 30% of your genome is homozygous, or 30% of your genes are homozygous, you're not going to get much map resolution around certain traits if these are in large blocks. And it opens up a potential of designing crosses based on which blocks are homozygous, and you can break apart that homozygosity in a targeted way and perhaps drive some heterosis in cassava. So there's some interesting implications there. So now we have 50 plus genomes. We have the ability to look at chloroplast DNA and nuclear DNA separately. So this is now an analysis of chloroplast DNA, showing the phylogenetic relationship between the samples. And a couple of things to point out here. First of all, Glasiovi is clearly very separate. This is this clade up here at the top, very different from the esculentas. Within the esculentas, we have some divergence about 100,000 years ago. And then the flabellifolias, which are the wild cassavas, actually have quite diverse haplotypes showing up here. There's a lot of different versions of this. But if you go down to the cassavas, there's only 2 chloroplast haplotypes in the entire 43 accession collection we have. And each of these haplotypes are found on 3 different continents. So this is a relatively old haplotype that has spread around since we've been spreading cassava around the world. One thing that's kind of unusual is that we have a Brazilian cassava here in blue that has a flabellifolia chloroplast, showing that there's definitely a lot of intermating that has happened in the past. If we go on to the nuclear genome, and we do a PCA analysis, this is from about 16 million SNPs from the whole genome data, we see 3 very clear clusters representing these species or subspecies, and a couple of samples that are in between. And so what we see here is that the tree cassava, this is that F1, and when I say tree, it's a very tall tree, not a small shrub like cassava. This is you're supporting the idea that this is an F1 between the 2 species. But we also have 2 flabellifolia samples that are kind of out of their cluster. One is clustering with the cassavas, and one is in between. So this one that's clustering with the cassavas has an has a flabellifolia chloroplast, but is behaving, its nuclear genome looks like a cassava. And this one here is slap bang in between the 2, and we haven't looked into that one yet. Another way of looking at the same SNP data is to do some population structure analysis. And here we ask for 3 groups, and these are how the 3 genome origins kind of break down. And a couple of things to point out here. So the Glasiovi species is very distinct from the rest, and this is the one on the end. And the flabellifolias are distinct from the esculentas. This is the tree cassava, so 50-50 of the 2 parent types. And then we've got some cassavas that have some Glasiovi origin. And this is something I'm going to drill in now. This only happens in African cassava, even though both Glasiovi and esculenta are South American in origin. So we only see evidence of this in African cassavas, of the ones we sequenced. And we only see them in lines that are derived from that Omani breeding program in Tanzania in the 1930s. Now remember, this was a deliberate strategy the Omani breeding program used, was to bring in some wild relatives. If we take that data and identify diagnostic SNPs for the Glasiovi origin, we get about 1 million SNPs across the genome that can be used to sort of diagnose a sample as a segment, whether it came from Glasiovi or esculenta. And orange here is the esculenta. Green is heterozygous for esculenta and Glasiovi, and blue is homozygous for the Glasiovi. So up here is the tree cassava, which we expect to be an F1, and we see it's green, mostly heterozygous, but there are 2 arms that are homozygous for Glasiovi, suggesting that this was actually when they crossed the Glasiovi with the esculenta already had some Glasiovi in it, and so when they made the hybrid, you've now got some homozygous arms. And then all of these lines here, the green, are Glasiovi segments that came in segments that came in Glasiovi from one of the parents. Now, all of these lines are Omani program derived. If we delve in a little bit closer and ask what's in common between all of these Omani breeding program lines, we get this one segment on chromosome 9 that is found in every one of the lines we looked at that derive from that program. And if we take the actual SNP haplotype within that segment, we see very clearly this has a single origin. This is the same source for this entire segment. So these lines are derived from the same original cross. And then lastly, we have GBS data from a much broader collection of African cassavas, and it shows that this segment is found in many more cassavas than we did whole genome sequencing on. Now, the question we haven't had time to answer yet is, does this indicate that it predates the Omani breeding program, or are all of these lines derived from Omani material? And so that's something we need to delve into. And the obvious question is, what does this segment confer? It's very tempting to say it confers disease resistance because that's what that program was all about, and that's what they were selecting for. But we have no evidence that that is necessarily where it is. What will help us point towards that is the real reason we did all of this work. And this is, our project was funded as a sister project to another project in East Africa, where the goal was to map different sources of Brown Street tolerance and then deploy or move that towards the ability to deploy marker-assisted selection to move tolerance around into different breeding programs. So where we are right now, there are 6 mapping populations as part of that project. Each one has had the parent sequence, has had the population GBS. All of that data was part of the data I've already talked about. Unfortunately, the phenotypes are not quite ready, so I can't tell you where these traits are mapping to, but we're going to find out very soon. And this is when these two projects will really come together and point to how genomics is really benefiting cassava. In addition, we believe the state of sequencing today could actually lead us much closer than just a linked marker if we have a region to zero in on. And so that'll be a potential follow-up to this. So just circling back to where I started, knowledge-driven crop improvement for cassava started with this, and now we've created some resources, okay? But what has happened, so what, you know? Well, there's actually new projects that are coming out that are starting to use these resources, and this is really the whole point of building a foundation of data. So on the biotechnology side, there's a project to create and engineer herbicide tolerance in cassava by specific site mutagenesis, and they're using the genome sequence to design that strategy. There's a funded project on engineering photosynthetic efficiency, which is mining the cassava genome data for orthologs in particular pathways. And on the breeding side, there's a whole host of projects, some doing QTLs, some doing association mapping, some doing getting ready to deploy marker-assisted selection. And then Gary mentioned Cassava Base, which is a database that's housing a lot of breeder-ready mapping data to feed into genomic selection models for increasing the rate of gain in cassava. Particular point I want to mention on increasing the rate of gain for cassava, it's really about increasing the speed of cassava breeding. The rate of gain, obviously, per year is going to increase, but cassava, because of the vegetative propagation, you get one plant from a cross, you get one seed. Then, if you want to do a yield trial on that plant, you now have to multiply it. Now, cassava, you can get about 5 plants from the stake per year. So you grow it for a year, you take it 5 stakes, and then you can multiply that times 5 for several years. So until it can take 4 or 5 years to get enough material to do a yield trial from your original cross. And with genomic selection, you can make a decision on that one plant. And so that's the point of genomic selection for cassava, is really to try and cut down that really extended timeline that cassava has to go through because of its genetics. One last point I want to make is that these data resources are all well and good, but what's really needed to use these is people who are knowledgeable and skilled in exploiting these. And so capacity building, in my opinion, is not so much in how to generate these data, but how to use these data. That's the most critical gap that, particularly in the Cassava community, we have today. So that's going to be our next focus. So lastly, I just want to thank all of the collaborators we have. Most of the genomic work was done in California, UC Berkeley, and Dan Roxar's group. We had some collaborators in the US that provided some material that they had on site. And then we have numerous African collaborators that provided us access to their material, spent many hours painstakingly doing mapping populations, and hopefully the data we've generated can be useful for them. So thank you, and I'll take a few questions.
Thank you, Steve. Does anyone have a question for Steve?
I was surprised to know that even outcrossing, this is having more homozygosity. So if deeply probed, this may be a model to other outcrossing plants.
Well, yes, so it is just conventional wisdom that is outcrossing. I mean, to me, I grew up in the Arabidopsis world where everybody knows everything about almost every second that an Arabidopsis plant lives and how it lives. That so little information is known about cassava, period. It was just really striking to me. And so I think there's plenty of room for explanations where we're just wrong about what we think cassava is. So I think there's going to be a lot of questions that we can explore with this data. So thanks.
Thank you.